Hi guys, welcome back to my channel. Let's do a biochemistry passage today. And then this passage is from AAMC section bank. Usually MCAT passage has 5 to 7 questions, but today in this video, we're gonna do the 3 most challenging questions of this passage. And you're also welcome to take a screenshot, to take a step at this on your own before we dive into this. We're gonna read the passage and do all 3 questions. And then the passage itself is not super challenging, but I think those questions are pretty tricky and they're really what set you up to be 520 plus. All right, let's dive into this and read the passage. So the passage reads, resistant hypertension is a type of hypertension that remains uncontrolled despite treatment with antihypertensive medicines. It is hypothesized that the signaling of prorenin, an inactive precursor of the renin component of the renin-angiotensin system in the central nervous system, plays a crucial role in the pathophysiology of this vascular disorder. Okay, so first paragraph is straightforward. It's just telling us that the signaling of a prorenin is responsible for this disorder in some way, right? Okay, let's keep reading. Researchers have shown that binding of prorenin to its receptor, PRR, okay, the receptor is called PRR, in nerve cells of the hypothalamus results in a conformational change in prorenin and activation of its catalytic functions. C17XA, a substrate analog which binds to the active site of the activated prorenin, has been used to inhibit the catalytic activity of prorenin. Okay, second paragraph, it looks short though, but it has a lot of information that's telling us that this prorenin is an enzyme, right? You can tell from the catalytic function wording here. And then also that we know this C17XA, this is about what? About enzyme inhibition, right? And then this inhibition is happening at the active side of the enzyme prorenin so that we can immediately know that, oh, this is a competitive inhibition, okay? All right, let's keep reading. To investigate the roles of prorenin and its hypothalamic receptor in pathways leading to resistant hypertension, researchers cloned the gene encoding PRR, wild-type PRR, and constructed several PRR variants. The dissociation constants Kd of prorenin for each of the PRR variants were determined. Okay, so basically wild-type PRR is just the original PRR, right? And then the PRR variants are some changes, some modifications they did to the gene. So let's see the table. So the table says dissociation constant Kd of prorenin for various PRR variants. Okay, so here are the variants, and then the first one is the one that's the wild type, right? It's the one that before any modification, but the other ones, as you see, that they have the modification to one amino acid, right? Like for example, this one, V109E. We have this amino acid, this protein is a long chain of amino acids, right? The polypeptide chain, and then the number 109 amino acid, it was a valine, but we took it out and then we replaced the valine with this E-glutamate, okay? That's what it means, just a modification of the amino acids, okay? All right, and then as we change modify the amino acids, that we see that different Kd, different dissociation constant, okay? The Kd really have some increasement here and then some decreasement here, okay? All right, so let's keep reading. Mouse neural 2A cells were transfected with either cloned wild-type PRR cDNA and an empty vector, okay, as control for 72 hours. Okay, so basically wild-type PRR is just a normal PRR, right? And an empty vector is the cell line that without PRR, right? You knock the gene out and then so you won't produce that protein, that enzyme, okay? All right, cells were then treated with a PBS buffer control and angiotensin II receptor antagonist losartan, prorenin, and a combination losartan and prorenin for 30 minutes. Okay, so everything I highlight here are basically what? Are the independent variables of my experiment, right? Things I get to modify, I get to control, if you will, right? All right, let's keep reading. The levels of reactive oxygen species, ROS, and intermediary molecule responsible for hypertension were measured. Okay, whatever is measured is our dependent variable of this experiment, right? And then you can also see that's on the y-axis, right? That's what we are testing for this experiment. Okay, perfect. So that now I move on to the figure, then I see, okay, these are the controls, and then this black bar is the empty vector without PRR, and then this gray bar is is the wild type PRR, okay? And then y-axis again is the ROS that we're measuring. And then higher ROS, the taller the bar it is, what does that indicate? That indicates higher hypertension, right? Because the ROS level is basically equal to hypertension level per passage, okay? All right. Let's also read the legend to make sure that we're not missing any of the information. So figure 1 says, relative level of ROS in neuro 2A cells for 3 days after transfection with either an empty vector, black bars, or wild-type PRR cDNA, gray bars, treated with various compounds. Okay, so various compounds are these, right? These are the various compounds. Okay, so, I mean, no new information from the legend, right? And I think we're ready to move on to the questions. Okay, so this first question says, based on the data presented in figure 1, and then I have a copy-pasted figure 1 here for you, okay? Expression of a PRR, 1 says increased blood pressure in part through an angiotensin II dependent pathway. 2 says increases blood pressure in part through an angiotensin II independent pathway. And then 3 says decreases blood pressure in part through an angiotensin II dependent pathway, okay? And then you're asked to choose 1 or 2 or 3, right? So, I mean, we briefly read about it, and I know that the choices, the differences of the choices are 1 and 2 are talking about increasement of the blood pressure, and then 3 is talking about the decrease of the blood pressure, right? So I want to look at my graph to see, my figure to see if the blood pressure is increased or decreased due to the overexpression of a PRR, okay? So let's look at this. So we see that the gray bar is having PRR, right? So this is PRR, we have a PRR, PRR, and then PRR, okay? Then all the black ones are what? No PRR, right? Empty vector, okay? Okay, so do you see all the gray bars are taller than the black bar or lower or shorter than the black bar? Right? Under any circumstances, under any treatments, even it's a control or losartan or prorenin or whatever, you always see that the gray bar is taller than the black bar, so that we know whatsoever the wild type PRR, aka the overexpression of PRR, is increasing our blood pressure, right? So right off the bat, I can cross out 3, right? And also C, right? Okay, so the next task is to figure out if it's angiotensin II pathway dependent or if it's independent of this angiotensin pathway, okay? So let's figure that out. Let's clean it up a little bit. Okay, so first off, let me actually make this, okay. Or sorry about that. Okay, so let's see if it's independent or dependent or probably both or neither, okay? If neither, then we don't have an answer to choose. So anyways, let's look at those graphs, okay? So this first one is control. Okay, fine. That's, I mean, without any treatment, right? This one I have PRR, this is without PRR. And then we see this losartan. And then what about losartan? If we go back to our passage, losartan is an angiotensin II receptor antagonist, right? So what is an antagonist? Antagonist means that losartan does bind to the receptors, right? But does it, after the binding, does the downstream biological reactions happen or not? Right? It doesn't. That's what an antagonist is, right? It does bind. It's kind of like an inhibitor in some way, right? I mean, you do bind to the receptor, but there's no downstream reactions happening. So basically, it's the blocking of this angiotensin II pathway, right? Okay, so the existence of losartan is basically to say no pathway, right? Or blocking the pathway, right? That's what it says. So, did I misspell it? So when you block the pathway, when you have no inference of this pathway, do you still see that the gray bar is taller than the black bar, right? You do see that, okay? So which means what? Which means the overexpression of PRR does increase blood pressure through an angiotensin independent pathway, right? So 2 is correct, all right? So should we just move on and then choose B, right? No, right? On MCAT, you always want to analyze every single choices, right? If you want to get a really amazing score. So we cannot just be impulsive and then choose B, right? We want to really examine one, right? It could be D, right? Okay, so let's see, what does one say? One says it's through an angiotensin II dependent pathway, okay? So how do we look at this? So the way we look at this is that we see that here, this 2 bars, with the prorenin, we see that, oh, with prorenin and with this expression of PR, we have a way higher blood pressure increasement, right? That's legit, because in the passage, the background knowledge is that prorenin is causing this hypertension, right? So that's legit, okay. But you can also see that here, with the losartan, right? When you block, once again, losartan here is no pathway, aka independent, aka block the pathway, right? So when you block the pathway, you see that the prorenin plus the expression of PRR, that one does not increase your blood pressure as much as this do, right? So that you would think, okay, losartan in this case, or aka blocking out the angiotensin pathway, does do something to the level of increasement, right? So this makes us realize that, okay, so this is also independent, right? So when losartan is playing a role, I mean, it also the overexpression of a PRR still increases the blood pressure, but not to a lesser extent, if you will, right? To a lesser extent, which means losartan is playing some role in this, which means angiotensin II pathway is playing some role in this increasement of blood pressure, right? It lowered the increasement, so that we will say, okay, but PRR still increased blood pressure, right? Despite that the losartan is in the way. So that 1 is also correct, right? So the best answer is actually D, okay? 1 and 2. I hope that makes sense, okay? This is not very straightforward. It's pretty challenging, okay? But I hope you see the logic, that you're always comparing the gray bar with the black bar to tell, okay, blood pressure is increased. And then you look at these two, that tells you that it's angiotensin II dependent, okay? And then some students may ask, okay, can I just see this and then tell that it's angiotensin II dependent? Isn't prorenin a precursor of renin, right? That indicated having this angiotensin pathway. Well, my personal opinion is I don't think so. I would think it's safer to compare these two to come to a conclusion, because the angiotensin pathway is the component, it's the renin, not prorenin, okay? All right, so I can't really say this prorenin treatment reflects a normal angiotensin pathway, okay? All right, next one. Based on the data presented in Table 1, which amino acid residues of PRR are involved in binding to prorenin? Okay, let's look at Table 1. All right, I hope you can see this. All right, so Table 1 is here, okay? Table 1 is here. Okay, so that we see the wild type has this KD of 0.034, right? And then the other ones, this V109E, this one has like a similar KD, right? But all the other ones, all these ones, they have a either higher or lower KD, okay? So first, the background knowledge you need to know about this is, what does KD mean, right? KD is an indication of affinity, right? So if your KD increases, right, it really means that your affinity decreases, right? That's an inverse relationship that we should know as background knowledge, right? So that you see that here, when I had all these amino acids, WDA, when I had all these amino acids, I would have this, right? Well, I will have the wild type without any change. But now I change the amino acids to L and K. That I have I have a difference of affinity, right? It either increased the affinity or decreased the affinity according to the change of the KD. Then you know that, okay, then I can tell that all these sites are where the binding happens, right? Okay, whereas this V109E, I mean, after I change the amino acid, it doesn't really change the affinity, means that it wasn't involved in binding at the first place, okay? So because of this reason, we're gonna choose residue 140, 140, 201, and then 269, okay? And then that will be D, okay? Okay, next question. Based on the data presented in table 1, which amino acid residue of pyridine most likely interact with the residue at position 201 of PRR, okay? All right, you can take a minute to think about this. Let me clean this up a little bit. Okay, so first we're gonna find where is 201, okay? So 201 is here. It goes from D to N, right? D is glutamate, and then, D is aspartate, and then what is E? E is asparagine, right? Okay, so we can see that as we go from D to N, I have increased KD, right? And then what does increased KD mean? It means a lower affinity, okay? So when you have this amino acid N, you have lower affinity, but the ideal case is when you have N, right? When you have D. When you have D, you have a higher affinity, right? You have a more binding. Okay, so the question is asking, based on the data presented in table 1, which amino acid residue of pyridine most likely interact with the residue at 201, okay? So for the pyridine to bind to this receptor, we want it to be D. We don't want N, right? Because the change of N increase the KD, decrease the affinity. So for pyridine to bind to this receptor, we want D. Okay, if D is what's existing in this active site, right? Then which amino acid, which answer choice would you choose? You think that would interact, would bind to D, okay? And then we know that D asparagine, D is asparagine, right? So aspartate, I'm so sorry. D is aspartate, okay? And then let's look at the other amino acids. So alanine has a really no charge, right? So we don't think it's gonna bind to aspartate, which is a negative amino acid, right? And then what about glutamate? Glutamate is also negative, right? And then what about arginine? Arginine is a positive. So yeah, C looks like a good answer, right? Because the positive thing will bind to the negative, our negative D, right? Okay, what about glutamine? Glutamine is also a polar uncharged amino acid, right? So the best choice is C, okay? So I hope that's helpful. These are not very straightforward, easy questions, but they really set you apart to be a perfect scholar. I hope that's helpful.